Hi everyone, it's Mr. Sinti, and today I want to talk to you about a really cool procedure in biology. It's called a transformation, and what we're going to do essentially is take DNA from one organism and actually put it into another organism and transform it, give it traits that it's never had before. If you recall our previous discussion, let's jump right into this. If you remember, we were talking before about the really cool experiment that the London physician Fred Griffith was performing in the late 1930s, early 1940s. Remember, he was working with pneumonia, and he was trying to find out what was going on with these mice, and he was injecting bacteria into the mice. And do you recall? He was using heat-killed S, which was the dangerous virulent strain, and he was mixing it in with living R, both of which don't hurt a mouse. But as it turns out, it did kill the mouse. And when he looked at the blood of the mouse afterwards, he determined that the R bacteria had become S. And so he used this word transformation, like they were transformed into something. In other words, they were able to produce a protein or a trait that they never were able to do before. And so we're going to sort of replicate that experiment. And this is a little discussion about how we're going to go about that. I think you're going to enjoy it. And so a little background on the organism that we're working with, which is E. coli. E. coli is your common intestinal bacteria, and it looks like this, sort of rod-shaped under the microscope. This is a scanning electron micrograph. And so some things about a bacteria that we would want to know is that they have a cell wall, kind of like a plant. They have a cell wall and then a cell membrane on the inside. And one of the things about a cell wall that's handy is that it prevents organisms from bursting when they're in a hypotonic environment. Do you remember osmosis, where if you had water, let me sort of illustrate this, if you had water out here, water would be going in and the cells would burst if they just had a cell membrane. But if they have a cell wall, that cell wall is very protective. And so you're like, why do I bring that up? It's that we've discovered that mold produces chemicals that actually kill bacteria because fungi are in competition with bacteria. So it was discovered that fungus produces these chemicals that we call antibiotics. And so what's really neat about antibiotics, you're familiar with those like penicillin, ampicillin, tetracycline. When you take antibiotics as a medicine, what it does is it doesn't hurt us at all, but it goes and it punches holes in the cell wall of bacteria. The truth is antibiotics work a couple of different ways. Sometimes they interfere with bacteria ribosomes, but in this case, let's talk about how they punch holes in the cell wall. And if they punch holes in the cell wall, that means that the bacteria will die in the presence of a high concentration of water. So they're very effective. We like antibiotics a lot. And so, as it turns out, some bacteria are resistant to antibiotics, and that means that they simply, when you administer antibiotics to them, they don't die. And so they have on their DNA genes that produce a chemical that sort of breaks down the antibiotic, and so they are not harmed in any way by this. And this is kind of scary. You don't really want bacteria to be resistant, as it turns out. And so some bacteria are resistant, and so they have genes, as I mentioned, on their DNA that codes for resistance. Like, for example, some bacteria are resistant to the antibiotic ampicillin or penicillin. And so here's a little diagram of a bacteria. They have one large chromosome where most of their DNA is. But some bacteria have these little circular sort of rubber band pieces of DNA with just a few genes called plasmids. And that's going to be a big part of our conversation about the experiment today. These plasmids, and this is an actual picture, this is a sort of a cartoon, this is an actual microscopic photograph of a plasmid. And interestingly enough, I was mentioning that there's some genes for resistance. And so let me just say from here to here is a gene that makes this bacteria resistant to ampicillin. And so I'm going to write that. I'm going to say positive. So it has a gene for this. And so, in other words, if I were to grow, this is going to seem a little bit weird, but if I grow this bacteria on a petri dish with agar, like this, and I place some agar in here, not drawn to scale because this is really big. If I have this bacteria and I sprinkle in some antibiotic and I'm like, ooh, I'm going to kill this bacteria and it's not going to be able to grow because I'm going to put ampicillin on here and it's going to kill the bacteria. They're not going to be able to grow. This bacteria will grow because it has a gene that is resistant to ampicillin. Okay? Just wanted to point that out. And so this is what it actually looks like. Here's a petri dish, here's some agar, and this sort of white material, these little white specks are colonies of E. coli. Those are not individuals, but rather millions of them. And you can see them even without a microscope. And so bacteria growing on ampicillin plates, that's AMP plate, it's agar plus ampicillin added. These are resistant, and so they have genes for resistance. Now, not all bacteria are resistant. Some are not, some are. And so the growth of the bacteria on an ampicillin plate sort of tells you that they are resistant. Here's my question to you. Do you think we're able to do this? You might say yes to this. Do you think we're capable of going into this bacteria right here? Let me just draw one of them. And let me just draw the plasmid right there. And it has a gene for ampicillin. Right there. Do you think, and I think you would say yes to this, we're capable of removing this plasmid right here from bacteria, and what we're able to do is put this DNA into another bacteria. Let me go a different color. So here's a different bacteria. Now let's say that this particular bacteria is not ampicillin resistant. I'm going to do AMP minus. Here's the gene. If we're able to put that DNA into this bacteria, in other words, like bring it over like this, make it take it in like this, it would then have the gene for ampicillin and it would live. And so we would have transformed it. We would have given it the ability that it never had before. In other words, it would be able to be living on an ampicillin plate. Now, you might be thinking, you know, it's one thing to be able to do something like that, to genetically transfer and genetically engineer a bacteria to become resistant to antibiotics, but why in the world would you do that? Well, you wouldn't do it just for this sake. There's another reason why you would do it, but I'm just pointing it out at the moment. And so, this is what we're going to do. We're going to take some petri dishes, either plastic or glass. In this case, we're going to use plastic, and we're going to make some agar. And the agar is not just going to be agar, but it's going to be Lauria bertini agar, which means that it has a nice mixture of salts and sugars, lipids, amino acids, vitamins. It's everything a bacteria would love to grow on. And so if we grew E. coli on a petri dish with LB, it would certainly grow. If we were to, and let me sort of illustrate that if I can. Oops. If there was agar in here, this is LB, like that, LB, and if we were going to try to grow E. coli on it, so here's E. coli. If we were going to try to grow E. coli on this, it would certainly grow, because all bacteria would love to grow on LB. You're like, okay, I can see that. But what would you say if we had some E. coli at the start? And it's like, where is this E. coli? Well, it's actually in a little tube like this. So it's a liquid like this. And so what would happen if we added ampicillin? If we added ampicillin to this agar, and it's a powder, sort of like a pill that you would take when you're sick, but you can crush it up like with a mortar and pestle and sprinkle it in when the LB is still a liquid, and then when it solidifies into a gel, it'll be mixed in. So it's like if we tried to grow E. coli on it and there was no growth at all, you would come to the conclusion that this particular bacteria was ampicillin minus. So this would be like a control. and say, okay, here's our E. coli at the start, that it can't grow on ampicillin. So if our goal is to take a plasmid that has ampicillin resistance and put it into E. coli, and then if it were to grow on this dish, we would say, hey, we have transformed the bacteria. And so let's bring up another gene of interest. Okay, so antibiotics are important as an indicator of transformation. I'm going to bring up something else as well, and this should seem familiar to you. This is the enzyme lactase, and the enzyme lactase is this protein can help speed up the breakdown of the disaccharide lactose. So lactose is a disaccharide, and it needs to be broken down, and the name of that enzyme is lactase. And lactase splits the disaccharide into the products are glucose and galactose. And so how do we know it's working? You can test for the presence of glucose or the presence of galactose. You can do that, but it's a little bit difficult to examine that. And so where I'm going with this is that some bacteria, you know how people are lactose intolerant, meaning they don't produce the enzyme lactase. Some bacteria are humorously lactose intolerant as well. In other words, they don't possess the gene for lactase. And so they're what you would say lac minus. And so this is something of consideration. If a bacteria is lac minus, we could actually give them the gene for making lactase and make them lac positive. It's like, okay, that's interesting. It's like, well, how would we know? If when we're using ampicillin, it's kind of easy to tell because they either grow or they don't grow. You can tell if you've transformed them. It's a little bit difficult to know if they're capable of breaking down a sugar or not breaking down a sugar. So scientists were thinking about this, and so they came up with this dimer, this chemical called, and it's a cool name, it's called pGAL. It's kind of sort of science fiction sounding, pGAL. And it's a dimer, and what's interesting about it is in the presence of lactase, the enzyme lactase, this sort of chemical called pGAL, which is right here, this is pGAL, it sort of acts like lactose. So when the enzyme lactase is present, it will hydrolyze and break that molecule down into two products. But curiously enough, let me overemphasize this, the products are really bright blue in color. And so if a bacteria, here's the take-home message, if bacteria are able to digest lactose sugar, and you grow them in a petri dish with the chemical pGAL, they will grow the color blue. It's remarkable because normally bacteria grow sort of a kind of a whitish color, but they will actually be blue in color, showing that they can break down pGAL, or they can break down lactose, because pGAL and lactose are sort of extremely similar to one another, simplistically speaking. So pGAL is an indicator whether or not bacteria can break down the sugar lactose. So think of pGAL as lactose, and the products are blue, so you can see it, and so it's easy. I mean, we want something that's easy, so we want to say, yes, it's growing, and it's blue. So that means that if they grew on a petri dish with pGAL and ampicillin, we could say two things. We could say that they're antibiotic resistant and they're producing lactase because they're digesting pGAL. And so this is kind of a picture of what they might look like. These are blue colonies, and this is sort of abnormal. This is not how bacteria really look. So these would be transformed cells. And this is growing on a petri dish, and I can mark it over here. This is a petri dish that has ampicillin. Of course it has LB. It has ampicillin and it has pGAL. And you can tell by the fact that they're growing that they're ampicillin resistant and that they can break down pGAL, and the reason they can break down pGAL is they're producing the enzyme lactase. And so in order to get the bacteria to grow, you have to, they grow very quickly. They often divide every 30 minutes or so. and if you give them a lot of good food, they'll be happy with that. But you do want to speed it up by growing them at sort of warm temperature, 37 degrees, which happens to be our body temperature. We can put them in a water bath, or we can put them in an oven incubator, and that's what we will do. So just a little refresher on this particular operon, the lac operon. You might recall that the way this works is there's a gene over here that produces lactase, and so RNA polymerase attaches to the promoter and it moves along and produces this. And so we talked about the fact that the lac operon is inducible, which means that in the presence of lactose sugar, or pGAL, this repressor protein will fall off, it will make lactase, and then digest the lactose sugar or the pGAL. And so this is kind of how we're going to roll with this. We're going to start off with a control, meaning that we're going to have E. coli. That's what this gray liquid is, the E. coli cells. And remember, I said that if we don't do anything to them at all, and we tried to grow them on an LB, let's jump all the way down here. If we try to grow them on an LB plate with pGAL, what would you think they would do? Do you think they would grow on just LB, just ordinary E. coli growing on LB? And you might say, yes, I think they would grow, and you'd be right. And you're like, well, what happens if I sprinkle in a little bit of pGAL in there? Would they turn blue? You're like, I don't know. Maybe they would, maybe they won't. If they grew and they were white, then you might come to the conclusion that, hey, they don't produce lactase. In other words, they're lactose intolerant because they're not blue. If you look over here at this petri dish, if you took E. coli, again, and you grew it on this plate, and it didn't grow, there was no growth at all, you would say, gee, I wonder why they didn't grow, they're dead. And you're like, well, there's ampicillin there, which must mean that they're ampicillin negative, meaning they're not resistant. So the trick is, can we make that same E. coli, we're starting with the same E. coli, can we make it to where they not only live on ampicillin, and they also make lactase? And so we want to put two genes into them. And the way we're going to do that in the experiment is, I'm just going to run through this fairly quickly. It'll be obvious when we're actually doing it. But the way we do this is simply take the E. coli, we add a plasmid that contains those two genes to it. We simply pipette that in there. We incubate it, which means just let it sit in a little ice bath for about 10 minutes, which chills them. We believe that the ice kind of slows their cell membrane down, and then we heat shock them. We take them from that cold and put them into a 42, which is pretty hot, almost lethal. and we're going to heat shock them for 90 seconds and then back on ice. And we believe what's happening is, inside here, the little plasmid that has ampicillin resistance, which is over here, this is the plasmid, has ampicillin resistance in the lactose operon. We believe that the cold, the hot, the cold allows it to go inside. And if it goes inside, now this bacteria, when it grows, and why is it going to suddenly grow? Because we're going to give it LB, which it really likes to eat, and then we're going to grow it in a 37-degree water bath for about a half an hour. And so the cells will grow and grow and grow like this, and this is the plasmid. And then what we could do is streak them onto a petri dish, and then the next day, if they grow and they're blue, we will know that we successfully transformed them. It's pretty cool. And so now the point of transforming a bacteria is not to make them antibiotic resistant or to make them lactose tolerant. That's not really the goal. The goal is to do something a little bit more clever. Like, for example, let me show you this. The goal would be if, for example, we wanted to make a protein that we really needed, like, for example, a protein called insulin. So if someone's diabetic, they need to have insulin. So there's a lot of individuals that have diabetes, and so we want to really mass produce insulin. Like, how do we do it? Well, there's a gene in all of our cells for the protein insulin. And so what we could do is we can take one of the human cells like this, and here's the gene that we're interested in, and this over here is a circular plasmid in bacteria. We can take an enzyme called a restriction enzyme. It's sort of like a little scissor, and we can cut a plasmid. And so this is what we do. We take plasmids from bacteria and we pipette restriction enzymes. They cut the plasmid open, and then they can also cut human DNA open. So do you see that little red area right there? That red area is, say, for example, the gene for insulin. And as it turns out that where it was cut is complementary to the plasmid, and so it's going to fit right into this rubber band. It's sort of like sealing it. And this enzyme called ligase sutures it in. And so now we have a plasmid right here that has human DNA into it. And so you're like, well, what's the point? What's the point of putting human insulin into a plasmid? I'm not getting it. Let me just pause this for a second and say, what would you say if we took bacteria, E. coli, and instead of just putting ampicillin resistance or the lac operon in there, let's say we put this plasmid into bacteria. In other words, we transformed it. What would this bacteria be capable of doing now that we put this plasmid in? And if you guessed producing human insulin, you would be correct. And so why would we want bacteria to produce human insulin when, how's that going to help anyone with diabetes? Well, E. coli divides very quickly, and it's one thing to make millions of it on a petri dish or in a flask, but if you grow a lot of E. coli, you can grow them in these big copper vats, like almost like in a factory, like a brewery, like for making beer. You could mass produce bacteria, and they just start making human insulin, and you're like, then you would crack the cells open and purify the human protein from all the other bacterial proteins. And it's actually not that tricky to do something like that. We can actually do something like that in the lab, purify proteins from bacteria. So it's a really powerful thing to be able to transform bacteria. Let's continue with this. And so if you're following it, we put a gene that we're interested in into a plasmid, and then what we're going to try to do is put that plasmid into bacteria and thus transform it. But here's my question. How do you know that this bacteria actually received the plasmid? How do you know it's actually going to be producing human insulin? And you're like, I don't know. Bacteria sort of just look like white. And you're like, well, yeah, unless we did what? Unless we could take this plasmid, and how would you like this? How about if we put, let's see if I can draw on that. Apparently I cannot draw on this. Yes, I can. How about if we put in the gene inside this plasmid for ampicillin resistance, so amp resistance, and we put in the gene for lactose. And so if you grew these bacteria on a petri dish like this, and it had some agar in it, and if these guys turned blue when they were growing, you'd be like, I successfully transformed these bacteria with the human insulin gene, and I know that because they're living on a petri dish with ampicillin, and they're living on a petri dish with they're living on a petri dish with pGAL, and so they're blue and they're alive. So therefore, the bacteria that we were starting with weren't able to do that, and so therefore we were successful at transforming them. So that's kind of neat. And bacteria would then be, you can streak them onto a petri dish, as I was just describing, and then they multiply very quickly, and they grow and divide, and so therefore you can actually see these colonies. And if the colonies appear, then you know that you were successful. It's kind of neat. Here's one last video sort of illustrating this as well. So you can take a human cell, crack it open. This is all the DNA from a cell. It doesn't even have to be human. It could be any organism. And then you cut that DNA with this special kind of restriction enzyme, sort of like the little scissors. And so you can cut DNA so that you can remove genes that you're interested in. And so it creates this sort of sticky end. And so here's the enzyme, cut. And so this is the gene that we're kind of interested in. Let's say that it's a gene for a growth hormone, for example, and we want to give growth hormone to individuals that are a little short. And so we can take that gene and then mix it in with plasmids. And now, so this is a circular piece of DNA that we've obtained from a bacteria. We cut that with the same restriction enzyme, and then we hope that that piece from the human that produces human growth hormone is going to insert itself into the plasmid. And again, we use this enzyme called DNA ligase. And so this technique is called a ligation. And let me pause it for a second. We actually perform this in our lab at school with our redwood DNA that we isolate and purify. We take redwood DNA and we ligate it into a plasmid, and then we transform bacteria with it. It's pretty cool. So all of these plasmids we would then put into the bacteria, and you're like, well, how would we do that? Again, we would put them on ice. We would then heat shock them, put them back on ice. And we treat the bacteria also with a little chemical, I haven't mentioned this before, a little salt that makes them a little bit more receptive to taking in DNA and induces them. And so how do you know that they've actually taken in the gene? How do you know that you're successful? What can you use to indicate whether or not you've successfully transformed the bacteria or not? And the answer to that is you would then streak them onto a petri dish, and if they grew, you would know that they have the desired gene. And so I hope you enjoyed that, and I hope it was informative. It might come to the point where you're not really following it absolutely until you actually do it, and that's fine, because experience is the best teacher, as it turns out. So thanks for watching.